This is part 2 of the lecture notes for chapter 3, and we had covered the carbohydrates, so now we're going to cover lipids. This is slide number 27. So lipids, we often think of as, fats and oils. There are some other examples, but they're large, non-polar molecules. So the carbohydrates are polar. They're, in fact, most of them anyway, especially the sugars. The monosaccharides and disaccharides tend to be non-polar. They tend to be polar, so they are soluble in water. If you just remember, you can dissolve sugar in water. So lipids are nonpolar. They're insoluble in water because being nonpolar, they're going to be insoluble in water. Water is a polar molecule. So lipids are not soluble in water. You probably learned this as a young person. Fats and oils and water don't mix. Anyway, functions, lipids are used for energy storage, but long term, whereas carbohydrates are used for like a quick energy source. Lipids can be structural components. They can help retain heat. They can be used for cell communication and regulation in the form of hormones, steroid-based hormones. There are some hormones that are protein-based, but then there are hormones that are lipid-based as well. And lipids can be for protection. So examples of lipids would be fats, oils, phospholipids, steroids, and waxes. You can see example of fats would be butter and lard. Oils would be like cooking oils. Oils are liquid at room temperature and fats are solid at room temperature. Phospholipids are found in the plasma membrane. Steroids are based off of the molecule cholesterol. So cholesterol is the precursor molecule for the sex hormones, testosterone and estrogen. Waxes are for protection and preventing water loss. So we have beeswax, earwax, the cuticle that covers plant leaves. All right, so lipids that are called fats and oils are triglycerides, and they're for long-term energy storage. They consist of one glycerol molecule linked to 3 fatty acids. If it is an unsaturated fat, then there are one or more double bonds between carbon atoms in the hydrocarbon chain. And the unsaturated fats are liquid at room temperature for the most part. An example would be plant oils like olive oil. Saturated fats would have no double bonds between carbon atoms. They tend to be solid at room temperature like butter and lard. And here you can see the hydrocarbon chain of a saturated fatty acid has no double bonds in it. And then the hydrocarbon chain of an unsaturated has 2. This particular one has 2 double bonds. And the way it's going to make the structure of the fat look is that the hydrocarbon chains are going to be completely straight and rigid in a saturated fat. Whereas in an unsaturated fat, they're going to be bendy and kind of flexible, and that is going to give it a liquid quality at room temperature. This is a glycerol molecule on the left and 3 fatty acid molecules on the right. The fatty acid on the bottom is unsaturated, and the 2 fatty acids on the top are saturated. So after a dehydration synthesis reaction, 3 water molecules are removed and the 3 fatty acid chains are attached to the glycerol. Now, to break this fat molecule up, that would require a hydrolysis reaction. So 3 water molecules would be added to separate the glycerol from the 3 fatty acids. Phospholipids are found in the plasma membrane or cell membrane, consist of one glycerol linked to only 2 fatty acids, and then also a phosphate group attached. So the fatty acids are the tails, of the phospholipid molecule. They are nonpolar and hydrophobic. That means they do not dissolve in water. Whereas the phosphate group is the polar head of the phospholipid, and it's hydrophilic, meaning that it is soluble in water. So the phosphate group, or the head of the phospholipid, always faces outside the cell and inside the cell, where it's going to be exposed to fluid. And the tails face each other because they are repelled by water. So phospholipids are found in the plasma membranes of cells, and here you can see the glycerol molecule and the phosphate form the head, and it is polar, so the head is soluble in water, and then the two fatty acid tails are nonpolar. So when you have in the plasma membrane, you have a double layer of phospholipids. It's called a phospholipid bilayer. So the tails actually face each other, and the heads either face outside of the cell, where they're in contact with tissue fluid, or inside of the cell, where they're in contact with the fluid that forms the cytoplasm. So the tails face away, and the tails face toward each other, and the heads face away. Steroids are another type of lipid molecule. They are composed of 4 fused carbon rings. Examples would be cholesterol, and then cholesterol can be modified to form testosterone and estrogen, which are sex hormones. Cholesterol is the precursor molecule for several other steroids. So you can see what they look like. This is cholesterol, a 4-ring structure. Testosterone has a few changes, but still a 4-ring structure. And estrogen, same thing. So cholesterol is the precursor for sex hormones, testosterone and estrogen. And cholesterol is also found in the plasma membrane, along with phospholipids. Then we have waxes, which are long-chain fatty acids connected to carbon chains containing alcohol functional groups. So a little bit of a difference in structure from the fats and oils and the phospholipids. Earwax, plant cuticles, and beeswax all contain wax. You can see the waxy coating on these grapes, and you can see beeswax here on the right. Now we're going to move on to the proteins. Proteins are polymers or long chains of amino acids linked together by peptide bonds. So amino acids would be the monomers. And down at the bottom, a protein is a polypeptide that has folded into a particular shape, which is essential for its proper functioning. So the polypeptide is really just a long chain of amino acids. And the protein is that same polypeptide that has undergone shape changes. So functions of proteins. Proteins have a large number of functions. First of all, for metabolism, proteins serve as enzymes. Enzymes are proteins that act as catalysts to speed up chemical reactions. Support proteins include keratin and collagen, and we have both of those in our skin and our bones. Collagen is found in our bones and our cartilage. Transport proteins include hemoglobin, which transports oxygen on our red blood cells. Defense proteins include antibodies. Antibodies are produced by B cells of our immune system and help us to fight against antigens, which are just molecules that stimulate an immune response in our body. Regulatory proteins would include hormones. There are some protein hormones like insulin that regulates glucose. And then there are proteins that contribute to motion, that contribute to contraction in our muscles. Actin and myosin are 2 proteins that cause our muscle cells to contract. Okay, so the building blocks or the monomers of proteins are amino acids. There are 20 different common amino acids, and they're all the same except for their R group. Their R group is the variable part that makes each amino acid different. So there are 20 different amino acids. That means there's 20 different R groups. But each amino acid consists of a central carbon bonded to a hydrogen, bonded to an amino group, which is NH2, and an acidic group, which is COOH. And these are the 20 different amino acids. I don't think it's all 20, but these are different amino acids that shows you different R groups, but all of them have a central carbon, an amino group, an acid group, and a hydrogen group. And then the purple shows you the R group. Okay, so if two amino acids are joined together, that occurs by dehydration synthesis reaction, and they're joined from the acid group from one amino acid is joined to the amino group of the other, and this is where that bond would be. This is a peptide bond that joins the acid group of one amino acid to the amino group of the second amino acid. And like all dehydration reactions, water is removed. And hydrolysis would be the reverse. We can break a peptide bond by adding water to it. So water plus this dipeptide here would break it up into two amino acids. So that would be hydrolysis. All right, as far as protein shape, protein structure determines its function. So when a protein loses its shape, it's said to be denatured. Certain chemicals, changes in pH or high temperature can denature a protein. And proteins that are fully mature can have up to 4 levels of structure: primary, secondary, tertiary, and quaternary. The primary structure of the protein is simply the sequence of amino acids that form the chain, that form the polypeptide. It can be hundreds, it can be thousands, it can be hundreds of thousands of amino acids that form a peptide, a polypeptide. The secondary structure is that same linear sequence of amino acid, either folded into a beta pleated sheet, kind of like if you ever made a paper fan, those pleats, or it can be in a spiral shape called an alpha helix. Alpha helix or beta sheet would be the secondary structure. The tertiary structure is the three-dimensional shape of the protein, and then the overall three-dimensional shape of the polypeptide. And then if you have more than one polypeptide and they are linked together, you would have quaternary structure. So here you can see the primary structure is just like a string of pearls. It's just amino acids connected in a chain. So these blue and pink balls are amino acids. And then the secondary structure can either be an alpha helix or beta sheet, as you can see here in pink. The tertiary structure is a combination of the alpha helixes and the beta pleated sheets, and they're just kind of all twisted into a three-dimensional shape. That's the tertiary structure. And then if you have this particular quaternary structure has 4 tertiary structures attached. So this is just a combination of more than one tertiary structure. And here you can see fibrous proteins like hair and the threads in a spider web. Okay, so sometimes proteins need help folding into their correct shape, and chaperone proteins will help the proteins fold correctly, and it also can correct misfolding. Prions are misfolded proteins that have been implicated in a group of fatal brain diseases known as TSEs. TSE stands for spongiform encephalopathy. I can't remember what the T stands for, but anyway, mad cow disease is an example of a TSE. So prions can cause neurodegenerative diseases, basically. Now we're on our section on the nucleic acids. Nucleic acid is the polymer, nucleotide is the monomer. There are two nucleic acids. There's DNA